I'm here now with Dr. Gagan Kumar of Northeast Georgia Health System. He works in the ICU in the health system, and he's also a researcher. I'm also here with Mark Meersman. He's the managing partner of IPC Global, a longtime partner of Qlik. Welcome to you both. What challenges did you have that made you seek out a software solution?
So at the beginning of this pandemic, there were actually more questions than answered. We didn't know what we're dealing with. This was a very different kind of a virus that was around, and getting a sudden surge of such a degree of sickness and mortality throughout the world, people were, including doctors, we were worried that we really didn't know much, and we wanted answers, but unfortunately we did not have them. The right answer was to let's do some randomized control trial with so many medication, but those things are going to take time. And what do we do in the meantime? And it's very difficult to wait for all the data to get collected, possibly, waiting for months before you have certain number of patients where you can make some meaningful connection. But trying to figure out ongoing patients' needs, if you can look at those data, if you're able to get that data and look at it and see how we are doing, makes a lot of difference. And again, one of the things in being a physician is you are not above the biases. You see one or two patients and something clicks, you start believing in it. But once you start looking at the data in much more detail, you can actually make more meaningful decisions without a lot of biases, which we have already seen in this pandemic. So many medications, so many things that have been tried, failed, people are optimistic upfront. So just trying to get a more scientific approach to the treatment and management, I think we needed somebody who could get us the data in a real-time fashion.
Thank you. Why Qlik though? What was it about Qlik that made us uniquely able to help you solve that problem?
Well, I would leave that answer to Mark, because when I met Mark for the first time, he gave me this information about the data collection. I knew that there were a couple of methods to get the data from our EMR, but it was very difficult for me to figure out like what is the best way to get those. And I think when I met Mark, he introduced me to this, how he can help me with this real-time data collection, graphs, trends. And to tell you the truth, I did not know that we could do that. And I think Mark can possibly tell us more about how the things really work at nitty-gritty details. But honestly, I would say it was mostly, I was lucky to meet him.
It was a unique moment in time, Gagan. We literally created two back-to-back applications that worked in tandem. One was called a population discovery. So Gagan and team could look at every patient historically and currently coming through the health system, through the hospital, through the physician practices, and see who are candidates for his study. Once we found that cohort, they call it, we then used the standard capability of on-demand application generation from Qlik to go gather literally every record ever created about these patients. So now we are talking about people that presented with positive and suspected COVID, and every piece of data, whether it was their labs, whether it was their medications before and after they came in the hospital, and all of the clinical treatments that were provided to those patients in real time. We reloaded the data every single day so that all the learning for Gagan and team could be cumulative on the fly. And so when I say every record, we're talking about hundreds of millions of records. The data was in near real time. The data was some structured and semi-structured. So we used some natural language processing to parse out pieces of the data to enrich it. We used tons of Qlik functions that are only available in Qlik that would allow us to prepare the data for statistics and machine learning. And all of that was done on the Qlik platform.
That's really great that you were able to pull all of that together. But what benefits have you seen as a result of bringing that all together? How has that impacted your work?
There were so, as I said, like there were so many questions that were up for the challenge, and typically starting with the simple things like when the pandemic started, the question of hydroxychloroquine. It was supposed to be the one of the panacea that was proposed to us, and it is very difficult when you're seeing patients in different time of your ICU rotations to see how those medications really work out. And that was just one medication. There were other things like steroids. There were a couple of other vitamins, zincs, and other supplements that people were trying to give these patients. And we were able to look at these to see if they were really making any difference. As we were getting more and more data, we were getting more confident that, okay, this is the thing that really is working, or this is the thing not working, and just trying to get above our biases. And just to make it more clear, I can possibly send you some graphs as well to see how we did with our oncoming data and how our prescription of medication changed over the past 1 year. It is just amazing. We are able to look at our stuff and whatever was pouring out from the other literature. And it's pretty interesting to see how whenever you look at the other people's experiences and your own experiences, you will see some conflicting results. But overall, the things become clearer with time as you get more patients treated with those conditions. So I think that it was pretty interesting. So, for example, I'll just take a medication called tocilizumab, and when this COVID struck, one of the hypotheses that was brought forth was, is it a pretty big cytokine storm? That's why people are getting worse and getting on the ventilator. And people started using this drug, which has been used in a very severe cytokine storms. And you can see when the patient came, our usage was pretty high. But slowly, as we learned more about this medication, more about this disease, we realized that we are overusing it. And looking at our data, we actually, by August or September, users had dropped down by like 40-50% to almost like 1 or 2%. And then we looked at more details, and we figured out that, well, you know what, this medication is working in one of those sickest of the sickest patients, which really works. And by October or November, we figured out that, okay, this medication actually makes some differences in lives of these people. And we are getting similar results from the other studies that were being published. And by January and February, we started using again, but now for more selected group of patients where it's supposed to work. So it was kind of very interesting to see how we evolved, getting a hypothesis, then not using it at all, and then kind of trying to understanding that, okay, this works, but only for people who have certain severity of disease. That is just one example. There are other examples like, hydroxychloroquine. There are new medications like ivermectin, vitamins, steroids. They have all seen this up and down in this pandemic, both depending on like what our data shows, what our experiences were, and how everything else from the world was coming through. So I think it was a pretty good way of looking at the data, changing and impacting your practices in real time.
So I understand you've put together a research paper based on the studies that you've done, and you're going to be publishing. Is that correct?
We have at least 5-6, and actually you can see the back of the screen. I have already written them out. Most of them are in submission phase, and this is one of the other interesting part of the whole medical thing is peer review takes time, and I hope that we can get this on pretty fast. But that's a very interesting thing, and we are knowing more and more about this disease every day. for example, we know that steroids work, but there's so many other questions. What kind of steroids do we need? How much doses we need? How long do we need to treat it for? That is the second stage of thing. And you can actually gather all this information in some form by looking at our data. So we are working on some of these projects, and we have found pretty interesting answers, and hopefully we'll be able to share with everyone.
Thank you. Mark, it's got to be, if it passes peer review, I'm sure you never expected to have your name published in a medical journal.
No, I had not. And it is a joy. In fact, as Dr. Kumar said, we actually have one published. I remember taking it home and sharing with my family over dinner the first paper my name was on. So thank you, Dr. Kumar, for including me and Drew, my colleague, on that. What I loved about what we did together is just that speed. Qlik wins every day on speed, and speed with quality engineered right into the process. We took a 7-step research process. We automated almost every step. So every night, as Dr. Kumar said, we were publishing new results, validating yesterday's findings, and yet asking the next question. There's a really important subtlety in the solution that we developed here. Is we brought in way more data, which is a feature of Qlik. We brought in way more data than we thought we would need. But guess what? When the next question came around, it was there, and the next question came around. So instead of saying, just pick what we need and we'll do that, we put what we need and some, so that everything that Dr. Kumar reported on had that context. And he was able to very effectively answer a question and answer the next question in the same breath. Very powerful.
So, just to give relevance to that, we are trying to figure out, well, this COVID infection, so much steroids, so much anti-inflammatory drugs that we are using, does it causes a lot of secondary infection? Because certainly those infections are also lurking in the corner as well. And with Drew and Mark, we kind of validated things, and every day we have new questions like, okay, we have know the type of organisms when they were really brought, but everything, okay, now maybe we have to make sure that they were real infections. So we have to make sure they had fevers, they had leukocytosis on the day that antibiotics were administered or the cultures were obtained. So trying to get into more nitty-gritty details and trying to validate those data as well. That was honestly, that was a piece of cake. These are very difficult to tease out manually from the charts, especially when you're looking thousands of them. But using a good algorithm, good validated algorithm, we are able to do it pretty accurately, I would say.
Well, excellent. Thank you. It's a great story, leveraging data to influence outcomes in healthcare. And I thank you for your time.
Thank you, Katie.